Good day, this is Professor Will, CBMD PhD. Today is Thursday, December 9th, 2021. It is 7:55 and 43 seconds, a.m. Eastern Standard Time. Today's research, immune cells. So let's hop to it here. Defense or repair. How immune cells are controlled during wound healing. Date December 8th, 2021. Source, University of Cologne. The scientists show a causal link between tissue repair, mitochondrial metabolism, and the activation and function of macrophages, scavenger cells. A Cologne-based research team has discovered that the metabolism of mitochondria, the energy suppliers of cells, in macrophages, coordinate wound healing to a significant degree. Macrophages belong to white blood cells and are also known as scavenger cells. The researchers showed that wound macrophages undergo different metabolic programs during tissue repair, which are required to support the successive phases for skin reconstruction after injury. In the human body, macrophages can adopt different activation states as pro-inflammatory macrophages in the early phase of wound healing. They kill bacteria or viruses and initiate a protective defense response. Reparative macrophages of late wound healing support the resolution of inflammation so that tissue can build up and equilibrium can be restored. An unresolved question in macrophage biology is what signals are required for the transition from inflammatory reparative macrophages. The research team demonstrated a functional link between tissue repair, cell metabolism, and the activation and function of tissue repairing macrophages. And changes in mitochondrial metabolism are the critical control mechanism for the different functions of macrophages during early and late wound healing. They found that sugar metabolism in the early phase is not sufficient to ensure productive repair. Using single-cell sequencing, the team discovered that some population of early-stage macrophages metabolizes reactive oxygen radicals produced in mitochondria as a byproduct of cellular respiration. The research team were able to show that for the first time, the benefit of reactive oxygen-containing molecules in early wound macrophages is essential to ensure the growth of blood vessels and thus timely healing. In contrast, macrophages use a different type of mitochondrial stromal exchange mediated by specific receptors, IL-4RA, for their anti-inflammatory and reparative functions in the late phase of wound healing. Based on the research results, it will be very interesting to understand whether disturbed mitochondrial stress in cells of the immune system contribute to aberrant inflammatory responses in the skin and pathological wound healing states. It will also be exciting to see a pharmacological intervention if mitochondrial stress responses provides therapeutic benefit and facilitates the repair of injured tissue. Good day, this is Professor Will, CBMD PhD, on microbiology, bioinformatics, and pathophysiology. This video was created for educational purposes only. Hope you enjoyed listening. Until next time, have a great day.